Hello there, you're welcome to Biology Lectures with Akirili Oladimiji Philip. It's my pleasure to have you with me on the lecture today. So today I'll be talking about oxygen-hemoglobin dissociation curve. This will be useful for all my A-level students, be it some Cambridge A-levels or medical students and what have you. Now, there's some basic facts I want us to understand. When you talk about oxygen-hemoglobin, this talks about the relationship between oxygen and how hemoglobin gets accepted by oxygen, or how hemoglobin gets saturated with oxygen, as the case may be. Of course, you know that the red blood cell is made up of this iron pigment called hemoglobin, of course. Specifically, oxygen binds to the heme group, which is a prosthetic group of hemoglobin as well. That's a type of protein. Now, looking at this graph here, you will see that the partial pressure of oxygen starts from, it is in millimeter mercury. It starts from 10, 20, 30, 40, 50, and what have you. And the oxygen saturation of hemoglobin, which you have right here, goes from 10 also, 20%, 30%, 40%, and it goes on and on. Now, you need to remember these few facts I'm about to establish. Hemoglobin accepts oxygen when there is high partial pressure of oxygen. All right? Anywhere there is high partial pressure of oxygen, that means around here, around here, around here, as I'm moving towards this, hemoglobin is going to be accepting oxygen more. For that reason, we say it has high affinity for oxygen. We can say affinity means acceptance, to accept oxygen. Now, on the other hand, anytime you have low partial pressure of oxygen, like you move from here towards this side, going that way, that means that hemoglobin will not be so ready to accept oxygen, rather it will be ready to release it. So we say it has low affinity for oxygen. So that means it's ready to release oxygen. The question you want to ask is, where in the body do we expect to have high affinity of oxygen? That should be in the lungs. Or where do we expect to have high partial pressure of oxygen? That should be in the lungs. Where do we expect to have low partial pressure of oxygen? That should be in the tissue. Why should it be low in the tissue? Sorry, don't mind my handwriting, please. Why should it be low in the tissue? Because in the tissue, you're expected to have high carbon dioxide. Why? Because there's respiration. Carbon dioxide will be released into the tissue because of respiration. It's going to release CO2 as a waste product. All right. Having established this fact, you might want to rewind the video if you need to re-listen to this part I've mentioned, these facts. Now, having established that, in your examination, you will be tested on this graph. You'll be asked to explain what does left shift means, what does right shift means. Anytime your oxygen hemoglobin dissociation curve shifts to the left, it means what? Higher affinity for oxygen. That means we expect that here we have high partial pressure of oxygen. So that means hemoglobin will be accepting here, accepting oxygen when it shifts to the left. Then also, when it shifts to the right, what does that mean? It means that there is low affinity for oxygen. That's hemoglobin having low affinity for oxygen. And what does that connote? It means it's going to what? Release oxygen to the tissue, which is what we want. Is this clear? Now, quickly, you might want to ask me, what are the factors that will make the hemoglobin or oxygen hemoglobin dissociation curve to shift to the right? Sorry, it's going to be low partial pressure of oxygen. shifting to the right. Then you can say high partial pressure of carbon dioxide. We call that Bohr's effect. Bohr effect means that presence of carbon dioxide or presence of high level of carbon dioxide causes hemoglobin to release oxygen to the tissue. So low partial pressure of oxygen, high partial pressure of carbon dioxide, high temperature. Let me write that. H means high. High temperature in the body. Decrease in pH. Decrease in pH talks about increase in acidity. All of these factors together will make your oxygen hemoglobin dissociation curve to shift right, which means it will cause hemoglobin to release oxygen. On the other hand, what can make your oxygen hemoglobin dissociation curve to shift to the left? That would be what? High partial pressure of oxygen, then low partial pressure of carbon dioxide, then increase in pH. That's alkalinity. When you have more of hydroxide ion, so to say, and lesser hydrogen ion, that is going to lead to high alkalinity. That will cause your oxygen hemoglobin dissociation curve to shift to the left. This is very important. You'll be asked to state what does left shift means. It means that hemoglobin is having higher affinity for oxygen, so it's binding to it. Then you need to remember the fact I just stated now that will make that to happen readily. All right, basically. So you should, if you trace this out, you go like that, you go like that. Now let me say it here. At 100 millimeter mercury, we know that your hemoglobin, oxygen hemoglobin association or dissociation curve will be around, or let me say your hemoglobin, or let me say oxygen will have saturated hemoglobin at 95% when oxygen partial pressure is 100. Let me take that again. When oxygen partial pressure of oxygen is 100 millimeter mercury, your hemoglobin will be 95% saturated. That's what that means. Basically, it's like more of like a law, so to say, under normal circumstances. All right. Hope this has been helpful. Feel free to rewind the video and send me your questions and your